Hello, my name is Toltelligent, and this video showcases the research done in the KU Ecology and Evolutionary Biology Department in the summer of 2015 by undergraduate researchers. Each student was told that they have 15 seconds to explain their research, although some completely disregarded this rule. After explaining their research, each student was asked a single question related to their work, and was asked to answer it as best as they could. Let's begin with the very well-prepared Sylvia Nunes.
Hi, I'm Sylvia Nunes, and I'm a student in Dr. Cartwright's lab. And this summer, I'm studying the evolution of salinity tolerance in an invasive cnidarian called Cordylophora caspia by acclimating them to various salinity treatments.
How will you be assessing salinity tolerance?
I will be counting the number of polyps, which are an indicator of colony growth, and using that information to determine the growth rate of each colony at various salinity treatments.
Up next is my own lab mate, Najoni Woody. We both worked in the Heilman lab. So, what are you working on this summer, Najoni?
So I'm working on a project that will help us better understand why and how a cycloidea-dependent program has been repeatedly employed across flowering plants to help make flowers with bilateral symmetry.
So how are you accomplishing your research this summer?
So I'm working with a species of tomato, which has radial flowers, and it has already identified cycloidea-like genes in its genome. So I will be employing PCR to check for expression of the cycloidea genes.
Up next is April Arkila, one of the two students working under Dr. Gleason. So, April, where is your research taking you this summer?
So I work with Drosophila biorhomipes. It's a species of fruit fly that's native to Asia. The males of the species have some black spots on the edges of their wings, and we're trying to see if the females of the species have a preference for males with these spots.
Have you turned up any interesting results so far?
We've actually found a lot of evidence to the contrary. It seems like the females don't really have much of a preference for or against the spots. Rather, it probably has something more to do with male aggression or competition that chooses who gets to mate.
Up next is Bonnie Cobb, the other one of the two students working under Dr. Gleason. What is your project for this summer?
So what I'm doing this summer is I'm working in Dr. Gleason's lab, and I am studying to see whether the novel beep component of Drosophila certivanti in their courtship song is an honest signal or not.
What is an honest signal?
So an honest signal is a signal produced by an organism where the quality of the signal reflects the quality of the individual that it's producing from.
Up next is Hannah Dornhofer. She worked in Dr. Brian Foster's lab. So, Hannah, what are you working on this summer?
I'm analyzing 30 years worth of tree growth data in an oak hickory forest at the western edge of its habitable range, so it's particularly sensitive to changes in climate and other disturbances.
Why is this work important?
Long-term studies like this are important because they help us understand how forests respond over long periods of time to disturbance, especially human disturbance like recent climate change.
Up next is Justin Jacobs. He worked in the reptile specimen collection in the KU Museum of Natural History. So, Justin, what form of research are you working on this summer?
I'm working in Dr. Richard Glor's lab in the KU Natural History Museum on species delimitation of Mantodactylus, which is a endemic group of frogs to Madagascar.
What makes this group of frogs special?
Well, in particular, Mantodactylus kalwani is a stream-dwelling frog, and they're in loud streams, and they don't call. Rather, they do foot flagging, and you can see bright markings behind their legs.
Up next is Natalie Vanette. She worked in the Blumenstiel lab. So, Natalie, what are you working on this summer?
I'm working in Dr. Justin Blumenstiel's lab, and I'm looking at the mechanisms of pi RNA synergism in Drosophila melanogaster that protect the fly against the harmful effects of transposable elements such as hybrid dysgenesis.
What are some of the symptoms of hybrid dysgenesis?
Hybrid dysgenesis causes chromosomal breakage, higher rate of mutations, and the main thing is it causes sterility due to non-functioning gonads.
Our next two students are Jeff Cole and Derek Harms, who are working under Dr. Jamie Walters. So, Jeff, what are you working on this summer?
I am studying the evolutionary rates of sperm proteins in monarch butterflies.
How did you gather these proteins, I guess?
I have soft hands.
So, Derek, what are you working on this summer?
We're looking at the gene products of female butterflies to find out which genes might be present on the female-specific W sex chromosome.
Have you found any genes so far?
So far, no. That'll be the final result of our project. But previous projects have found that there's usually very few, maybe just 1 or 2 genes present on the W chromosome.
Our next student, Jacob Peterson, was working in the rumen lab this summer. So, Jacob, what have you been working on this summer?
I'm looking at species in the North Atlantic Ocean, trying to create a taxonomic hierarchy and also a food web to try and see if species more closely related on both of those different variables are also related in how they're affected by climatic change and variables associated with that.
You've mentioned before that you've been using R in your work. How's that experience been? Have you used R before?
I really didn't ever learn R until I came here this summer, so it's been quite a big learning curve for me, trying to learn all the new syntax. I've done other coding languages in the past, and it's just been trying to see how to exactly create the right script for it.
Our next student, Rebecca Flournoy, was working in the Billings lab this summer. So, Becca, what are you working on this summer?
I am looking at if disturbance of an ecosystem's vegetation and the surface soil horizons has a long-lasting influence on the transformation of organic matter in deep versus surface soil.
What ecosystems are you taking your soil samples from?
We are looking at sites from hardwood forest, pine forest, and annually cropped cotton.
As for myself, this summer I'm studying the genus Penstemon, with special focus on the several independent switches in pollination syndrome that have occurred within this genus. What techniques did you use to accomplish this? I used a fluorescent staining technique to highlight the cell walls of petal cells in Penstemon flowers. Then I used a confocal microscope in conjunction with a 488 nanometer blue light to cause that dye to fluoresce in order to image it easily. Thank you for watching this short film about our research, which was made possible by NSF Grant DBI 1262795. If you are a fellow research student and would like to watch a fellow video on this channel that contains some advice and what to expect during your first research experience, that is available for viewing as well. Thank you.